What are we gonna learn today? So before that, I introduce you to my group member, Mina, Michelle, Muhammad Arif, Muhammad Abdul Ghafur, Munisha, Natia, Nameshwar Rao, Ang Jui Chun, and Nitya, and I myself, the presenter, Muhammad Umar Al Muiz bin Muhammad Zain. The topic today is Shistosoma Haematobium. Clearly, we can see the picture show us the histopathology of Schistosomiasis Haematobia. Histopathology of bladder shows eggs of Shistosoma Haematobium surrounded by intense infiltrates of eosinophils. Maybe some of us is still unfamiliar with the term Shistosoma Haematobium. So what is Shistosoma Haematobium? Shistosoma Haematobium is a urinary blood fluid, a species of digenetic trematode belonging to a group genus of blood fluid Schistosoma. It is found in Africa and the Middle East. It is the major agent of the most prevalent parasitic infection in humans. Schistosomiasis is an acute and chronic parasitic disease caused by blood fluid trematode worms of the genus Schistosoma. So let us begin with the discussion of the epidemiology of the Schistosoma Haematobium. Schistosomiasis is prevalent in tropical and subtropical areas, especially in poor communities without access to safe drinking water and adequate sanitation. It is estimated that at least 90% of those requiring treatment for schistosomiasis live in Africa. There are two major forms of schistosomiasis, intestinal and urogenital. The table shows the parasite species and geographical distribution of schistosomiasis. There are 4 species of intestinal schistosomiasis. The first one is Schistosoma mansoni. It is distributed at the Africa, the Middle East, the Caribbean, Brazil, Venezuela, and Suriname. And the second one is Schistosoma japonicum. The distribution of it is at the China, Indonesia, and the Philippines. Followed by Schistosoma mekongii. It is distributed at several districts of Cambodia and the Laos People's Democratic Republic. And the final one is Schistosoma genesis and related Schistosoma intercalatum. It is distributed at the rainforest areas of Central Africa. At the urogenital schistosomiasis, there are only one species, and it is Schistosoma Haematobium. Most of it is located at Africa, the Middle East, Corsica, it is France. We are going to look about the mode of transmission. People become infected when larval forms of the parasite are released by freshwater snails. It penetrates the skin during contact with infested water. Transmission occurs when people suffering from schistosomiasis contaminate freshwater sources with their excreta containing parasite eggs, which hatch in water. In the body, the larvae develop into adult schistosomes. Adult worms live in the blood vessels where the female release eggs. Some of the eggs are passed out of the body in the feces or urine to continue the parasite's life cycle. Others become trapped in body tissues, causing immune reaction and progressive damage to organs. So for the upcoming slide, we are going to look about the life cycle of schistosomes in humans and freshwater snails. Hope all of us are prepared. This is the diagram that all of us need to memorize, so it might help us during the examination or maybe when we answering the question about the life cycle. It's about complete life cycle in both humans and freshwater snails. Human is the definitive host and freshwater snails is the intermediate host. Definitive host is the organism wherein the parasite reaches its mature form and reproduces sexually, while the intermediate host is the organism that harbor the sexually immature parasite to undergo development and complete its life cycle. It releases eggs to urinary tract which excrete along the urine. So this is the freshwater snail life cycle. Life cycle of Schistosoma Haematobium in freshwater snails, which is the intermediate host, which going through the asexual reproduction. In stagnant freshwater, the eggs hatch into the larva called myracidia, either male or female. The myracidia pierce through the soft skin of the snail and move to the liver. Their cilia is cast off and extra epithelial covering forms within 24 hours. The myracidia transform into sporocysts Mother sporocyst produces many daughter sporocysts through active cell division. Each daughter sporocyst forms new larvae called scaria. The sporocysts rupture and scaria are liberated. Free scaria penetrate the liver and move out of the snail into water. Each scaria has a biforked tail that finds a human host. Scaria attach themselves on the skin using suckers. Start piercing the skin by secreting proteolytic enzyme that widens hair follicle. Tails removed during penetration. Head part enters. It enters the blood vessel known as cystosomula. It enters the systemic system to reach heart and liver. From the liver, they enter the portal vein to reach different parts of the body, reach the vesicle vessels through anastomic channels between radicals of the inferior mesenteric veins and pelvic veins. After living inside small venules in the submucosa and wall of the bladder, they migrate to the perivesical venous plexus to attain full maturation. Going to look about the clinical features. Schistosomiasis progresses in three distinct phases: acute, chronic, and advanced disease. On its early manifestation, swimmer's rash caused skin penetration by Schistosoma scaria. Acute manifestation, which is acute schistosomiasis, known as Katayama syndrome. Symptoms may include fever, malaise, myalgia, and fatigue, and non-productive cough, diarrhea with or without the presence of blood. The chronic and advanced stages, the obstructive diseases in the urinary system due to eggs trapped in the tissue. Secondly, hepatosplenic inflammation. Third, the granuloma formation and the local inflammatory response mediated by CD4 plus T helper 2 lymphocytes helped facilitate the passage of eggs into the lumen of the gut or urinary tract. Fourth, neuroschistosomiasis due to migration of adult worms to the brain or spinal cord. Fifth, gastrointestinal and liver diseases due to eggs retained in the gut wall, which can induce inflammation, hyperplasia, ulceration, microabscess formation, and polyposis. The above picture shows the clinical manifestations of acute, chronic, or advanced schistosomiasis. In these slides, we are going to look the laboratory diagnosis. The first is parasitology, the detection of Shistosoma Haematobium eggs in urine specimens. Second is by immunology, the serological test. Schistosome antigens or antibody detected in blood or urine samples, example, the ELISA test. Next, by filtration technique, using nylon, paper, or polycarbonate filters is the standard diagnostic technique. Children with Schistosoma Haematobium almost always have microscopic blood in their urine, which can be detected by chemical reagent strips. Some indirect diagnosis also can be done, such as radiological and also cystoscopy. Here are some of the list for the references that we used. For more details and information, you guys may get through these references. Thank you.